I'm David.
I'm Max.
And I'm Joanne.
And we at Neutralize will help you engineer happiness and cognitive performance in your experience. All right. Thank you, Steven Roodenrys, for coming on the Neutralize podcast.
You're welcome, David. It's nice to speak to you.
To start with, would you please tell us a bit about yourself, your history, and what you do?
Yep, sure. I'm a cognitive psychologist, so I study cognitive processes, and I've been doing that for, over 30 years now, I suppose. And I've looked at normal cognitive processes in terms of trying to understand how they operate, particularly in relation to short-term memory and long-term memory to some extent. And I've done a number of things as well of looking at where those processes might go wrong in groups like children with ADHD or with reading problems. And I've also become involved in research looking at ways of improving cognitive function. And I've been doing some stuff over the years with herbal supplements and foodstuffs with a group of people at my university over time. Mm-hmm. And yeah, that's basically, my sort of history.
Right. This concept you mentioned of short-term memory, I think that most people have an intuition of what it is, but there is a difference between it and some other concepts that are commonly used in cognitive psychology, right? Such as working memory. Can you please tell us a bit about the difference between working memory and short-term memory?
Yep, sure. Well, firstly, I suppose one thing is what most people think of when you say to them short-term memory isn't what psychologists mean when they're talking about the cognitive systems of short-term memory and working memory. Most people, I think, if you say that term, will think that you're talking about what's happened in the last day or two, when really by short-term memory, what cognitive psychologists mean is more or less just what you are aware of now, and it's really only what's been happening in the last half a minute maybe. Everything else is regarded as long-term memory, and in terms of the function and the sort of distinctions we can make, that's the best way in which we can separate memory up temporally in terms of the temporal period that it refers to seems to be what's just been happening in the last 20-30 seconds against everything that's gone before that seems to be a functional distinction. Right. So short-term memory is used to really just refer to the storage capacity that we have. Historically, it was sort of viewed as a record of the recent past of what's just happened. Some of the eminent early psychologists linked it to consciousness and regarded it as part of the mechanism by which we perceive time passing, and that may not be so commonly held now, but certainly it's a record of what's just happened. But over time, we've developed ideas that, initially it was seen as a single storage device, and then it's been seen that we actually have multiples that are linked to our sensory modalities. So we seem to have a short-term memory that's to do with what we've heard, and a visual short-term memory as well for what we've just been seeing, and probably, short-term memories for things like touch and taste and so on as well. Short-term memory was seen as being fairly crucial to cognition. It was seen as a sort of bottleneck for cognitive processing, in that it was about how much information you could keep active in your memory at one point in time. That idea was developed into this more recent notion of working memory, which was really about how memory storage capacities might be used in ongoing cognitive processes. So when you're asked to do, a little bit of arithmetic, or when you're listening to somebody speak and you're having to remember what they were saying at the beginning of the sentence in order to integrate that with what they're saying at the end of the sentence to get the whole meaning. Right. So that storage capacity in service to ongoing cognitive processes was seen as working memory. So the distinction is that working memory is really about holding and manipulating information or processing information at the same time. Short-term memory in the traditional way in that it was looked at earlier was much more just about simple storage and not about how it might interact with processes. So that's a sort of fairly simple sort of description, I guess, of what the difference is.
Could you give us an example of when
sure. So
yeah.
One of the traditional tasks for measuring short-term memory or researching short-term memory is the serial recall task, which is exactly like if I was to give you my phone number by just saying a series of digits to you, and you then had to write it down or put it into your phone or whatever, right? Mm-hmm. So that task of just holding on to that set of items, in this case the digits, and being able to reproduce them in the same order immediately afterwards, that's a stereotypical short-term memory task that's just tapping into your storage capacity. Right. A working memory task would be, I already mentioned, like arithmetic. If I was to give you a sum to do in your head, to say, well, what's 13 times 22, and ask you to work that out, you have to be able to remember the numbers that you've been given. You also have to be able to operate on those numbers. You have to remember any intermediate products and then put those together to get a final sum, and you have to keep track of which numbers are playing which role in all of that calculation. You've got to keep them in the right place while you're doing it. So working memory is much more about interacting with the information and manipulating it.
Right. I know you've done some research on different words and how they are kept in short-term memory, or how they're stored in short-term memory. What aspects of words make us remember them more or more likely to?
Well, for short-term memory, it's dominated by systems that are involved in remembering the sound of the words. Okay. So, we can talk about words being represented, by their sounds or by their meaning. And of course, when we're using words, normally those two things are very closely tied together. Mm-hmm. But you can also hear and repeat back, a nonsense word or something. So when you hear a new word, for example, a word you've never heard before, then you are able to hold on to that word even though it has no meaning at that point in time, right? And you may in fact be trying to work out what the meaning of this word might be given the context that it's been used in. So what is this word referring to? So the phonology or the sound of the word is represented separately from the meaning. And of course, normally we develop representations of both aspects of the word and they're closely bound together. But in a short-term memory task, the way most people do it seems to be more heavily reliant on just trying to remember the sounds of the words that you just heard rather than processing the meaning in a very deep way. Whereas in working memory, it tends a bit more, and certainly in long-term memory, it's much more about the meaning, that you're processing the meaning and remembering the meaning of the words. And working memory tends to be a bit more of a mixture of the two, a bit more emphasis on the meaning and not quite so much on the sound.
Right. And this is how you measure the effect of different interventions on short-term memory and working memory as well, right?
Yes, that's right. So we have tasks to measure those sorts of capacities. So I mentioned before the serial recall task, which is just to present you with a sequence of, say, 5 or 6 items, and at the end for you to say them back in the same order. So most people can do that with maybe 5 or 6 short words. When you make the list a bit longer, 7 or 8 or 9 words, then they can't do it anymore and they start to make mistakes. So you're able to measure the capacity of that system, and you can obviously see, if it differs under different manipulations that you might make. With working memory, the tasks are often ones where it interleaves processing and presentation of words that you are asked to then recall later on. So one of the first tasks that was sort of used in this area was referred to as sentence span, where people had to read a sentence and then make a judgment about whether that sentence actually made sense or not. And it might be a sentence where that doesn't make sense. So, the banana stood on the deck of the ship, and then they had to remember the last word of the sentence, and they'd read another sentence and remember the last word of that, and read another sentence, remember the last word of that, and then have to recall the words those last words in the order in which they read them.
I see.
Right. So it's drawing on this capacity for both processing and storing at the same time.
Yeah. So you use these sorts of tests both to study which words, for example, are very, or rather are more likely to be recalled, such as words with meaning for long-term and working memory, right?
Yes. So one of the things that we investigate to try and get a better understanding of how memory systems work is to manipulate the nature of the sort of stimuli or the words that we're using in the task. So some of the work that I've been involved with, particularly over the years, has been looking at how the representation of the words in our long-term memory systems might influence performance in these short-term memory tasks, and what that tells us about the extent to which these systems are really separate. So one of the big debates in the history of psychology is about how many memory systems there actually might be. And some people have sort of claimed over the years that we have a short-term memory system that's very much like the RAM in your computer, that you copy things into the RAM by typing in on the keyboard or by retrieving it from the hard disk, and then when it's in RAM, it's highly available for processing. And it's a discrete copy of what might be in the long-term system.
Right.
And if that was the case, then there shouldn't really be any influence of long-term memory factors on how well you can remember those items. So, for example, one of the basic findings is that you do better at remembering words that are more common than words that are less common. And in the short-term memory system, that shouldn't happen if what it's doing is just putting a copy into RAM and leaving it there.
Right.
There's no reason for that to differ depending on, how often you've heard that word in the past. That effect has to be arising from how the words are represented in the long-term system.
Right.
So that's one of the sort of things that I've done in my career in trying to investigate aspects of the normal memory system in humans.
Right. And when you have a short-term memory, let's say a RAM activation through being exposed to a set of words, for example.
Yes.
Then that retrieval of information from long-term memory is also a sort of problem-solving cognitive task, right?
Yes, it can be. So one of the tasks that is used a lot in the research literature around memory and neuropsychological function, and it's used a lot in the research investigating nootropics and dietary effects and things, is to read a list of about 15 words and ask the person to just recall as many of them as possible. That task draws on a very wide range of aspects in the brain. So it draws on language areas and it draws on frontal lobe areas, because people are trying to work out different cues they can use to try and remember the items. So when you've just heard the list and you're trying to think back what was it that I just heard? You're trying to create a cue using your frontal lobe system to probe some of the memory systems that are, further back in the brain. And so it's quite complex and it taps into a range of areas in the brain.
Yeah. Speaking of nootropics, I know that you've at least read quite a bit and are quite knowledgeable about the history of use of cognitive enhancers. Could you please give us a quick recap of how people have used cognitive enhancers historically?
Yeah, sure. I wouldn't say I was an expert on it, but I've done some research on a Ayurvedic herb, and I've sort of looked at some of the traditional Chinese medicine approaches. So yeah, one thing is clearly in traditional medical practices like Ayurveda coming out of India and traditional Chinese medicine, there's a long history, going back thousands of years, looking at herbal materials, to improve cognitive function. Often that's about memory, but at times, about other aspects of cognition. Again, sort of frontal lobe control, executive function to some degree. Western society probably had the same sort of history, which has sort of been a bit lost or suppressed, I guess, by the development of more conventional Western medicine. But I'm reasonably sure that there would have been traditional herbal preparations for cognitive problems in Europe as well. Certainly in more recent times we had widespread use of stimulants.
Mm-hmm.
So a lot of the sort of drugs which became illegal were at one point legal and used as stimulants, of course. The well-known story that Coca-Cola supposedly originally contained cocaine.
Right.
And various things, I suppose, tobacco and coffee, obviously extremely widely used for a very long time. And is probably the most widely used stimulant in the world now. And
right.
So people have been using it to improve cognitive function, both in that short-term type approach of something like caffeine to sort of give you a bit of a perk up for a short period of time, but also in the sense of more as a medicinal treatment to try and improve a chronic problem.
Yeah, I want to touch on both of those later in this discussion, so acute and chronic effects. However, I'm interested currently, right now, how is the use of cognitive enhancers distributed over the population as of 2020?
Well, I did a review of the literature on this a little while ago. It's getting towards, a little while ago for a book chapter that I was writing. And there's not really any research that's directly asking about the use of nootropics or herbal supplements or supplements in general, specifically for cognitive improvement. There's some focus on the use of stimulant drugs, so particularly things like Ritalin, which is used as a medication for ADHD, being used by college students, for example, to improve their cognitive abilities. But there's not a great deal of other research that really is directly trying to look at the use of nootropics. It comes out a little bit in broader studies that are just looking at supplement use in general. But those sorts of studies are just sort of suggesting that, you've got 30 to 40% of people using supplements for one thing or another, and mixed in amongst that, of course, is the ones who are taking it for cognitive benefit.
Right.
It does seem to be split from what we can see in the studies that are picking up on some of this. It seems to be split quite a lot between students using it to often just to be able to study better, to perform better at academic tasks. That's often done in a way which is, you might say, sort of trying to compensate for other behaviours that might be having an impact. Right. So, trying to cram for exams, or, pulling an all-nighter is the expression, to get an assignment finished on the night before it's due.
Yeah.
And so using stimulants to stay alert and continue to do what they hope is good work. So that's one group who use it fairly heavily. The other group that seem to be using cognitive enhancers more is older people who are trying to, resist cognitive decline with age. So there seems to be an increasing number of people who are interested in taking supplements in order to maintain cognitive function, as well and for as long a period as they can.
I know a lot of studies have been done on that population, elderly, trying to.
Yes, it is a function. Yeah, it's a focus for research as well, seemingly, that to look at older people and whether we can improve memory function, because we know memory function starts to decline quite early in adulthood. So for a lot of cognitive functions, in fact, they peak really probably in your 20s, and most start to decline from then on. So there's a natural decline. And then on top of that, of course, you get people with more of a pathology-related decline in memory and cognition, generally, later on, talking sort of 60s and 70s. And so it's an area where people can see that, well, if performance was better in the past, can we recover some of that lost function, and delay the further decline in cognitive function, and particularly in relation to diseases like Alzheimer's, where it is something where it's got a very obvious pathophysiology underlying it, which we don't fully understand, but it sort of raises the possibility that we might be able to, have an intervention that would improve things for people who are suffering Alzheimer's and maybe slow down the rate of decline.
Right. How useful is evidence that something does work for people with age-associated memory impairment?
There's some evidence for some substances.
Right.
It's, in my view, there's not very many things that have a good evidence base. There's a lot of studies that are being done or have been published. There's a lot of inconsistency in the literature as well, though. So, some of the studies you might be able to find, well, yes, there's a handful of studies that have been done on a particular substance or a particular herb, and they might all report an improvement in some type of cognitive function, but across the studies, you might not see a consistent improvement in which particular cognitive functions, have changed. And to me, then that evidence is not all that convincing, because if there is a mechanistic explanation for an effect of a particular substance, then you would expect a degree of consistency. You would expect those particular cognitive functions to be improved in all of the studies that involve that particular substance. So when you look at the research literature and you see that it's not as consistent as you would expect, then it doesn't increase your confidence that those effects are genuine.
Right. I guess the problem is that people are so different, and when you study as you have to, and I think it's great that you do, but when you study substances and their effects on a large group of people, it's going to be hard to notice effects that would appear if that substance was perfect for a specific individual.
There's always individual variation. So yes, so when we run a study and we give, let's say 50 people a supplement, there's always a range of changes. So some people will show quite a big change at the end of the intervention, taking the substance for 6 weeks or whatever we're doing. Other people will show very little change. Some people will actually get worse.
Right.
And that's the nature of what's going on in the research, right, is that if you give a substance where there is no effect, some people will still perform better on the second time than they did in the first time, right? But that improvement can't be attributed to the substance.
Yeah.
Right. And so that's what we're looking at all the time in the research, is that we know that we're working in an environment where some people are going to change due to other reasons and their performance might go up. Some people's performance might go down for other reasons. And what we're trying to see within this larger group is a sort of signal that within the group as a whole, there is an added positive effect on top of this sort of noisy pattern of change across all these different individuals, right, that is suggesting a consistent effect of the substance. So if we have enough studies with enough people in them, then if a substance has a genuine effect, we should be able to see it fairly reliably. But still within that, that's going to be demonstrating that if you have enough people, enough of them will show an impact, a benefit from this substance, if it does have a benefit, for you to be able to detect. Right. So you should be able to see that, but you will always still then have individuals within that sample of people who will respond more than others. Right? And there does seem to be individual variation in the biochemistry that influences the extent to which people are affected by these things. The body's very complicated. The processes involved in ingesting these substances and them getting through the digestive system and broken down into metabolites, which it might not be the substance itself. It might be one of the metabolites that it's being broken down into that actually has an effect. And that it's affected by, in some cases, the microbiota, the gut flora, the makeup of the type of bacteria that are in your gut can influence how much of this substance is going to get through into your bloodstream,
right,
or how much of it's being broken down before it gets in and what it's being broken down into. So you get individual variation for all of these sorts of things.
Right.
But if there is an effect, of a substance, if we have studies with enough people, we should be able to see it consistently.
Right. I was talking to Emily Leproust from Twist Bioscience yesterday, which is a company working on genetic solutions, essentially. So they enable people to sequence their genomes, for example. Do you think that it is a promising field of personalizing medicine so that we can, based on the individual's biochemistry?
Oh, absolutely. I think it's a promising field. I think the question is more about how far off is that promise.
Right.
We're learning more, and more genes are being identified, the role of particular genes, and which particular processes they might influence and so on are all being identified. But that I think a lot of that is focused on diseases, understandably.
Right.
And so, most of the genes that are being identified that I'm aware of are more related to, processes that result in, an illness or something I would consider a disease or a fairly serious problem, as opposed to genes which result in variation that is still within what you might call the normal range.
Right.
That we all vary from each other in all sorts of different ways. And, we can think about that as just normal variation, so that, some people are better than others at different things. Some people's bodies process particular substances more efficiently than others. But as long as you are reasonably healthy and intact, we regard that as just normal variation. And that variation probably has a genetic basis. But those genes, I think, not so many of those are being identified yet as to, how they might interact or relate to cognitive functions or drug interactions and things like that, drug effects.
Right. I was speaking to Joe Cohen from Self Decode on another podcast a couple of weeks ago, and they are trying to deliver such a product where you can upload your single nucleotide polymorphisms. Are you familiar with that? Yeah. So essentially gene variants, and
yes,
and they are trying to help you based on your data.
Yeah, well, I don't know this area particularly,
right?
So I couldn't say how many of those things have been mapped, I guess. My feeling is that there's a lot of work going on now, but I'm not sure of how long it's going to be before those things are really useful.
Right.
For most people, I don't know.
Right. But something that I'm pretty sure you do know of is Bacopa Monnieri and its uses for improving cognitive function. Will you tell us a bit about your study that you did in 2002?
Yep, sure. So Bacopa Monnieri, or Brahmi is the term for it in Ayurvedic medicine, has been used for thousands of years as a memory tonic, is a phrase sort of that's used. It's a weed that sort of grows in waterways, all around India. I ran that study on it because I had a student who was working in disability support, who was working with people with brain injuries, and they were seeing it advertised specifically by somebody who had an acquired brain injury, who was attributing their recovery to taking Brahmi or Bacopa. And they were asking her, whether they should try taking it. And she sort of came along to me and asked if I knew anything about it. And I said, well, not really, but I've always been curious about some of these herbal things. And she sort of said, well, could we do a study? And so with her and a few other students, we ran that study with the support of the company that were producing the product. We approached them and they were happy to be involved and supplied the product, and also arranged for us to get a placebo. And the result was the paper that you're referring to, where we did get an effect on memory. Mm-hmm. We tested a number of different cognitive functions, and this is one of the things about the way in which these studies are done. And the sort of statistical analysis that's done. I'm not sure how much you understand about the hypothesis testing and the significance testing.
How much I understand about it?
Yeah, so all your listeners, I guess. But
yeah, exactly. And please keep it quite basic. Yeah.
Yeah. So one of the things with these sorts of studies is that um, with traditional statistical testing of the results, um, we take an approach which basically evaluates um, more or less sort of the chance that these results that we've got actually are coming from um, a situation where really there is no effect. All right. So maybe that's the way to talk about it is it's basically saying what chance is there that the data that you've got has actually come from a situation where there is no effect. And if that chance is relatively low, we decide, okay, well, then that suggests that this effect is real.
Right.
Right. So if you test enough cognitive functions in the one study, then there's a likelihood that you will get one difference or more differences purely by accident, when really there is no effect. It's just that by random chance, you've got what appears to be, an effect of the substance.
Mm-hmm.
So in this particular study of ours, we did what's typically done of testing a number of different cognitive functions, and we did only get one or two significant effects, which does raise the possibility that that effect is just an accidental finding, right? And really there is no difference.
Right. How many cognitive functions did you test?
Well, I think from memory we tested about 10.
Okay.
So there was a reasonable chance, but probably more tests than that actually in that paper. Mm-hmm. So there was a reasonable chance that it was just accidental. The one thing that gave me a bit more confidence about it, though, was that the one that came out being significant is the one that I would have predicted as being most likely to show an effect based on the studies that had previously been done with rats. Mm. So of all the cognitive functions, the ones that most closely map onto the sorts of manipulations and things that they look at with the rats, that particular one was the one that came out as being significant when most of the others didn't. And then, of course, over time, the other thing that builds confidence that it is a true effect is that other people report studies that show the same effect on the same type of task or the same cognitive function.
Right. What was that effect?
So in that case, it's actually the task I was referring to before, where you are presented with a list of about 15 words to remember, and then have to try and recall as many as you can once the list has been completed. Mm. And then the same list is presented again, and you attempt to recall it again. So generally, a normal adult would be able to recall maybe 5 or 6 words on the first presentation. And then as the list is repeatedly presented and they try and recall it again, they add to their recall, so that by the end of 5 or 6 presentations of the list, most adults would be getting close to all of the 15. So they might be getting 12 to 15 of the words correct. So you learn through the repeated presentations. And that task is the one that in our study particularly showed an effect and has shown positive effects in other studies with Bacopa Monnieri. And interestingly, it's also the task that seems to show an effect most consistently across the literature with all sorts of different substances. So of all the different things that I've seen people investigating memory function in different sorts of memory functions, different memory tasks, visual memory tasks, short-term memory tasks, this task I'm talking about is called auditory verbal learning, and it's regarded as a long-term memory task. Because it involves this sort of learning over time and repeated presentation of the list. And then it's usually administered in a way that you do 5 presentations and recalls, and then 20 minutes later, you ask the person to recall the words again. And so it's a long-term memory test at that point, particularly because you had no mention of this task for 20 minutes, and often you've had them doing other cognitive tasks in between. And so if performance is better at that point, then that's an effect of long-term memory. And it's the task that seems to most consistently show an effect across all the different nutritional intervention sorts of studies that I've been looking at.
What do you think that is?
Well, I think it probably relates to what I was saying before, that it draws on quite a wide range of areas in the brain. And I think that perhaps that it's just a little bit more sensitive than a lot of the other tasks as a consequence of that. And effectively, it's just better able to pick up more subtle changes than some of the other tasks. But it may be that it's specifically, linked to particular aspects. So, for example, that task as a memory task draws on areas of the brain like the hippocampus. And the hippocampus is one area where we know where the brain generates new neurons. And so it may be that, with chronic supplementation, what you're doing is having some sort of impact on, the number of new neurons that are being created in the hippocampus.
Right.
So, that would be a fairly specific effect linked to memory, and, wouldn't necessarily affect other cognitive tasks like attention or visual processing, for example, because they don't draw on the hippocampus to the same degree.
How transferable are findings?
That's always interesting. In the study of ours that you referred to, we recruited. We wanted to avoid people who had dementia, so we recruited people who were under 65 years of age, but sort of into the later middle age. And we specifically tried to recruit people who felt their memory wasn't as good as it used to be. And we asked them about their memory function, both before we started the study and after they'd been taking the Brahmi, so at the end of the study. And on those sort of more subjective measures of memory performance, we didn't really get an effect. So on the sort of what you might think of as your everyday memory function, people didn't feel that it had improved particularly, but they did better on the objective measure that I've been talking about. Now that sort of disconnect there might be because a lot of people, when they think about their memory function and whether their memory's very good or not, are not really thinking about their ability to learn new information, which is the sort of task that we were talking about. What they're thinking of more, I think, often is two things in particular, I think. One is remembering things like people's names, so people that they know, and certainly I think everybody as they get older has this problem of seeing somebody who, you know who that person is and you know quite well, and yet you can recall other information, but you can't recall their name. Right. And word finding difficulties is another one that sort of seems similar to that in, you can't think of the word that you want. That again, that's a relatively common thing that seems to be something that increases in frequency with age. So those sorts of things about accessing information that's already in memory didn't appear to change in our study. And the other thing that I think a lot of people think about in relation to their memory in the real world is about remembering to do things. So remembering that you have to pick up milk on the way home, or, that you have to make a doctor's appointment or take your medication, those sorts of things. Right. And again, that's a different sort of memory, and it's not about storing new memories, encoding and storing new information, which is the sort of task that we did show an improvement on. So the Brahmi studies, suggest that it might be helpful for students studying and trying to learn new things. Right. And I have had a couple of students actually anecdotally tell me that they took Brahmi particularly when they were doing subjects that involved a lot of rote learning. So sort of learning neuroanatomical terms and things like that, where they felt that it benefited them. But again, that's just sort of anecdotal, but it matches up with the experimental data around the type of task that we would expect them to show some improvement on.
So Bacopa Monnieri is for learning, and that memory functions related to that task.
Yes, so it seems to be helpful in creating new memories for material, but not necessarily for helping you retrieve information that is already stored in your memory somewhere.
Right. Yeah, that goes hand in hand with my anecdote as well. I think you've read it on our blog.
Yes.
I had great results with Bacopa Monnieri for my, I'd say, working memory, but also the effect that you just talked about. So learning and really recalling information that I learned maybe a couple of hours ago or a couple of days ago.
Yes, yeah, that's exactly the sort of thing that the experimental sort of evidence would line up with.
Mm. You mentioned rat studies earlier.
Yes, although it's a long time since I've read those, and I'm not sure I can remember much detail of them.
I see. Let's move on to a more broad question. And how, or rather, which nutritional interventions to improve cognitive function have the most science backing their efficacy and safety?
Well, in terms of memory, I think Ginkgo Biloba and Brahmi are probably the two that have the strongest evidence. None of them, in my view, on reading the evidence, there may be a few recent papers that I might not be familiar with, but from my reading of the literature, there aren't any that really have an acute impact. So, if you just take a heavy dose immediately, it doesn't have a benefit. Mm-hmm. The only sorts of substances that do that are stimulants, so like caffeine, which really just sort of maybe improves your alertness and influences the way you encode information. So it's sort of peripheral to memory in a way, in that it's not really having an impact on the storage of the information in your memory, but it's probably allowing you to maintain your concentration and encode the information better in a way in that case. Right. Glucose can have an effect. So there are studies where supplementing or giving somebody a dose of glucose will improve function. Though the studies that show an effect of that tend to be the ones, though, where people have actually been not eating, so they've been fasting for some period of hours before the study, so that in a way it might be more just that dose of glucose is returning the body to its more normal functioning level. So I think there are some studies that maybe show a bit of a boost. But yeah, again, that's sort of just, in relation to things like glucose and the stimulants, but there's not really that improved memory as such from a single dose. Or there's not very many studies, and not enough to be convincing at this point for me.
Right.
There are other studies, though, suggesting long-term effects. So I've already mentioned Ginkgo and Brahmi, obviously. I've been involved in some nutritional research with my colleagues here. And we've been looking at a group of chemicals called anthocyanins, which are the chemicals that give fruits and vegetables, they're dark purple and red pigments. So they're quite common in strawberries and blueberries and some of the dark-fleshed plums, and a few other things. And there's a few papers out there now suggesting a benefit from dietary supplementation with anthocyanins, taking, or eating, consuming, reasonably large quantities of those sorts of things, of berries.
Right.
So the studies we've been doing have been, drinking 200 mils of juice, for example, every day. Some of the studies that have been done in animals, so again in rats, that have shown positive effects have, been giving the rat an equivalent of something like asking a human to eat, a whole punnet of strawberries or a whole punnet of blueberries every day.
Right.
So yeah, those ones are also, I think, showing potential for some benefit as a long-term supplement. But it's difficult, of course, because those things are seasonal. They're relatively expensive. Processing can destroy those chemicals. So if the food being processed, so if it's being stored, if it's being turned into a juice or something like that, the industrial processes on those can actually destroy the chemicals, and make it, so that really there's none of that chemical still active or available in the substance. Mm-hmm. So there's a lot of work still to be done in trying to work out how foods can be used other than, through fresh foods.
Right. I know there are extracts of blueberries, for example, containing standardized amounts of anthocyanins.
Yes. Yeah. So there are people who are, working on and have developed, I think, means of, as you say, extracting without destroying the anthocyanin. My understanding from my colleagues in nutrition is that, the sort of heat processes that are generally applied to foods destroy the anthocyanin, and there's then no benefit from consuming it.
Right.
So yeah, companies are aware of that and are working on ways of preserving it, and extracting it in a way that's going to still be available within the supplement.
Right. You mentioned caffeine and glucose for acute cognitive enhancement earlier.
Yep.
In my experience, both of those have negative effects if I use them consistently. So if I drink several cups of coffee in a day, every day, then I build tolerance and perform worse at baseline, for example. Yeah,
absolutely. That's the pattern with many drugs is tolerance. So you get a reduced effect.
Yeah.
The more often you take it, the smaller the benefit becomes, and so people take more and more of it. And then, of course, even with you get the standard idea of drug withdrawal, you get that with caffeine as well. So people who've been drinking something like 8 cups of coffee a day, if they stop taking caffeine, will feel pretty terrible and, may suffer headaches and other sorts of problems. And many of those things seem to be that they sort of induce a subjective improvement, and then, of course, that declines as the drug wears off. But that subjective improvement might actually just be, taking you back up to what would be your normal level if you weren't using it.
Right.
So once you've become a habitual sort of user, it might be more that that substance is actually just effectively getting you out of the slump from when you're not dosed up on the caffeine. I've seen studies that suggest that that's the case with nicotine with smokers, that really what's happening when they're smoking is just that it's removing the craving and bringing their system back up to a more normal level of functioning, rather than giving them a boost to their function over and above what you might think of as a normal level.
Right. You mentioned long-term neurogenesis in the hippocampus earlier. Of Bacopa Monnieri. That is not a mechanism that is happening in nicotine, caffeine, and glucose, right?
No, absolutely, yeah. No, so yeah, those sorts of things are having short-term effects on neurotransmitter levels and things like that, rather than more chronic effects related to you might say the structure of the brain. I suppose if you're talking about neurogenesis, you're adding new neurons and they're making new connections, and you're changing the function of your brain.
Right.
You're allowing it to function in a better way.
Right. Would it be reasonable to assume that herbs that do improve hippocampal neurogenesis and increase it, that you don't build tolerance to them, and I guess specifically Bacopa Monnieri?
Well, again, that's a really interesting question when you start thinking about the human body and the diversity and the complexity of the interactions that are going on, and that the human body is, a set of homeostatic mechanisms. So when you start increasing the levels of availability of one substance, it may actually trigger a process that acts to reduce the levels of that substance in your bloodstream. Right. So it may be that you can get some benefit initially, but it may be that benefit declines over time. It may be that for some people that homeostatic mechanism works particularly efficiently and removes excess amounts of those chemicals or those metabolites, etc., from the bloodstream before they really manage to do any good. So it's all very complex, and difficult to predict exactly what's going to happen for anybody in particular.
Right. And I think it's great that there is so much new research coming out on all of these underlying mechanisms of how the body absorbs and distributes drugs and how they bind to different receptors.
Yeah, absolutely. There's a huge research going on. It's all very complicated and difficult to get your head around all of it and stay on top of it.
Right. Is there any specific research that is exciting to you, that's ongoing or is going to happen?
No, I wouldn't say anything particularly that I'm excited about. I guess I'm a little bit skeptical about the benefits for people who are well nourished and healthy. Most of the studies where you're seeing effects tend to be in groups, like as we said before, older people who've got declining function.
Right.
You might be able to improve that, or groups who've got deficits of some type. So for example, we know and have known for many decades, right, that iodine deficiency results in a lack of development in parts of the brain.
Mm-hmm.
And that's a nutrient deficiency. But if people have a normal level of iodine in their blood and you supplement them with iodine, it doesn't result in even better, development of the brain.
Right.
And again, it's one of those sort of homeostatic mechanisms, I think, that the body sort of removes the excess of what it doesn't need of these things.
Right.
So I think for a lot of the research, and it's not clear also, like with the reporting about subjects or people who are participating in these studies, there's often no reporting around what their ordinary diet is like.
Mm-hmm.
And so, they live in, most of them are obviously done in Western societies where the population's relatively well nourished. But even within the Western societies, we have a clear thing about people not eating enough fruit and vegetables, and so they're not getting enough of the micronutrients. So it might be that some of these effects that we're seeing are really because some people in the particular samples who are taking part are, a bit deficient in their diet, and really that this is just sort of helping to bring them up to a more optimal level, which would be obtained if they just ate a healthy diet anyway.
Right.
So, without that information, it's hard to say. But basically, I think there's a lot more research needs to be done before we can really be confident about any of these things having a notably positive effect on, memory function.
Right.
I know you talk in your blog about, things like healthy lifestyle effectively, getting good nutrition, getting enough sleep, getting exercise, being physically healthy and so on, right?
Yeah.
So all of those things are important, and, it may be that the benefits we see from supplements are going to be more beneficial to people who haven't got that sort of healthy lifestyle, and that there are processes that are not working in the way that they're meant to or at their optimum level as a result of those lifestyle factors. It's those people maybe who are going to show benefits from supplementation.
Yeah, that makes sense. If someone wants to read your studies maybe or come in contact with you, where can they find you online?
Well, I'm at the University of Wollongong in Australia. So people can email me. It's Steven with a V, then _ Roodenrys, ROODENRYS, at uow.edu.au.
Great. Thank you for coming on the podcast, Steven.
You're very welcome, David.
All right. Thank you very much for listening to this episode of the Neutralize podcast. But please remember not to take anything that we said as a substitute for professional medical advice, diagnosis, or treatment. And before you start experimenting with nootropics, please get the basics of biohacking right. So that is your sleep, your exercise, your nutrition, and mindfulness. They are going to get you much farther, much faster in terms of optimizing cognitive performance, happiness, or any other goal that you may have with your experience in engineering. But when you want to start experimenting with nootropics, our app will be a great place for you to start. Based on hundreds of placebo-controlled human studies, we give you a nootropic recommendation personalized for you. So all you have to do is go into the app at beta.neutralize.com, link in the show notes, and enter the effects that you're looking to get from a nootropic. You can add up to 3. So let's say you want to improve your mood, you want to protect your brain, so neuroprotection that is called, and you want to improve your attention. So you add mood, neuroprotection, and attention to your search for a nootropic. And then you click search and you'll have a nootropic recommendation at your hands based on hundreds of human placebo-controlled studies that we've gone through for you. And we'd love it if you leave a review on this podcast with some honest feedback. And other than that, thank you very much for listening once again, and see you next time.